Hey guys, so today I wanted to talk about DNA replication. And so the DNA replication has many steps to it, but the ultimate goal is for it to go from one strand of DNA to two strands of DNA. And so the first thing that happens is it needs to unwind. And instead of going to like a massage parlor or putting the feet up or watching a nice video, what happens is the way DNA replication, or the way DNA unwinds, is it uses helicase. Helicase is a enzyme that goes through the DNA, and it actually breaks apart the bonds between the two nitrogenous bases. So that would be either the AT bond, adenine and thymine, which has their bonds are formed between hydrogen bonds. So they have two hydrogen bonds between them, whereas cysteine and guanidine, they have three. So you have helicase, which breaks apart those hydrogen bonds, and that temporarily causes the DNA to go from two strands to being broken apart, so you get two individual ones. Okay, and next thing that happens is you have single-stranded riboproteins, single-stranded binding proteins, and what those do is like their name says, they bind to each of these strands, and they stabilize them. Because if those weren't there, those exposed nitrogen bonds and the phosphate backbone, which is heavily negatively charged, it's going to be really destabilized in the cell, and that can lead to it could potentially kill the cell or rupture the cell. Next thing that happens is after these proteins bind to each of these strands, you have synthesis that occurs. And so really the process of synthesizing a new strand of DNA is you have to add a primer, and after the primer has been added, you have DNA polymerase, which will bind to that primer and start synthesizing. It just adds a bunch of nucleotides that are complementary to the strand that is coding or non-coding, depending which side. Next thing that happens is we'll start off with the leading strand. So leading strand adds nucleotides in the 5' to 3' direction. That means the strand itself is going 3' to 5'. So when the primer binds to it, with the 3' to 5' end, when the primer binds to the strand, what happens is DNA polymerase will bind to that primer and just start adding nucleotides. Okay? Now, contrariwise, we have the lagging strand. So the lagging strand is going 5' to 3'. So when the primer binds, it's going to be going in the opposite direction. And it only does this through fragments. And these fragments that occur are known as Okazaki fragments. Okay? And so after the Okazaki fragments are formed, what needs to happen is there needs to be a way to bind the Okazaki fragments and the remainder of the growing strand. And so the way the enzyme that is used is a ligase. Ligase is very important because it binds the Okazaki fragment and the growing strand together. They form a bond between the 5' phosphate and the 3' OH of each of the respective Okazaki fragment and the growing strand. Okay, so this is DNA replication, and I ran through it really quick. If you guys have any questions, comments, or concerns, please email me at gforbes@poets.whittier.edu or comment below. Thank you.